You know, for all of human history, extinction has been a one-way street, a final chapter, an absolute ending. But what if it wasn't? Right now, technologies like cryopreservation and gene editing are really starting to blur that line. They're challenging the very definition of gone forever, and they're forcing us to ask some huge questions about the difference between life, memory, and extinction itself.
So, let me start with this. A story about a goat that went extinct twice. Sounds totally impossible, right? How can something go extinct more than once? Well, this strange, kind of unbelievable paradox is the perfect way to kick off our journey into the really complex and controversial world of de-extinction.
All right, so let's meet this twice extinct animal, the Pyrenean ibex, also known as the bucardo. This mountain goat's wild ride to the brink and back, and then, back again, is this incredible scientific drama that shows us both the amazing promise and the huge challenges of this whole new frontier.
So here's how it all went down, kind of like a four-act play. First, the original extinction. The very last bucardo, a female named Celia, died in 2000. But, and this is key, before she passed, scientists took some of her tissue and froze the cells. Fast forward to 2003, and they actually did it. They created a clone from those cells, genetically identical to Celia, and it was born. But tragically, it had a severe lung defect and couldn't breathe. It only lived for a few minutes. I mean, think about that. It was a groundbreaking success and a totally heartbreaking failure, all at the same time.
The Bucardo experiment, even though it failed, just blew the doors open on this massive philosophical debate. It forces you to ask this question. If a species' unique genetic code, its DNA, is sitting safe and sound in a lab freezer somewhere, is that species really extinct, or is it just on pause?
Well, the people who are really pushing for de-extinction have a pretty radical term for this state of being. They call it evolutionary torpidity. The whole idea is that as long as the genetic blueprint exists, the species isn't gone for good. Its evolution is just kind of paused, waiting to be rebooted. And look, this isn't just playing with words. It's a fundamental shift in how we think about what conservation could even mean.
So how in the world do we get to a place where we could even have this conversation? The answer is really in a technological revolution, one that taught us how to literally put life on ice, creating these modern-day Noah's Arks of frozen genetic material.
It all happened surprisingly fast. Back in 1949, scientists figured out that glycerol could act like a kind of antifreeze for cells, protecting them during freezing. Just 4 years after that, boom, we get Frosty, the first calf ever born from frozen semen. It completely changed animal agriculture. And by 1975, conservationists caught on and created the very first frozen zoo. All of a sudden, the goal wasn't just to save living animals in parks, but to bank their genetic code as the ultimate insurance policy.
So once we had that technology, this really ambitious vision started to take shape. People started asking, what if we could do more than just store DNA? What if we could actually use it to right some of our past wrongs and give nature a second chance?
And, proponents of this idea will argue we've already done a low-tech version of this. Just look at the peregrine falcon. By the 1960s, it was pretty much wiped out in North America because of the pesticide DDT. So conservationists brought it back by breeding some closely related subspecies and releasing them into the wild. The bird we see flying around today isn't a perfect genetic copy of the original, but it fills the exact same ecological role. It works.
And that brings us to a much bigger, broader definition of de-extinction. It's not always about making a perfect one-to-one clone. For groups like Revive and Restore, it's about bioengineering a living animal to fill the hole that an extinct species left behind. So the focus really shifts from genetic purity to functional replacement. Does it do the same job in the ecosystem?
And the vision here is, well, it's huge. We're talking about grand ideas, like restoring entire lost ecosystems by putting mammoth-like creatures back in the Arctic tundra, or engineering disease resistance into species that are on the brink, and maybe, just maybe, replacing some of our collective guilt over extinction with a new sense of wonder and active restoration.
Okay, so that's the dream. But this incredible promise comes with a whole lot of peril. Bringing species back from the dead forces us to face some of the most complicated ethical questions of our time. I mean, are we just being innovators here, or are we crossing a line? Are we playing God? The whole debate really boils down to this: promise versus peril. On one side, you have the promise of restoring nature. But critics, they raise some really serious flags. For example, does bringing back woolly mammoths create a moral hazard? Does it make us care less about saving the elephants we still have? And what about the money? Well, these super flashy, headline-grabbing projects suck up all the funding from the less glamorous, but still vital conservation work. And maybe most troubling, does this technology just give a few wealthy donors the power to decide which species get a second chance at life?
And let's be clear, this isn't some abstract futuristic debate. A few years back, a biotech company announced it was bringing back the dire wolf, and it landed them right on the cover of Time magazine. The message to everyone was loud and clear. The impossible is happening right now.
But here's the reality check. Almost immediately, other scientists pointed out that these weren't actually dire wolves. They were modern gray wolves that had been genetically engineered to look like dire wolves. And this gets to the absolute heart of the controversy. This isn't really about pure revival. It's about bioengineering new kinds of creatures that we then choose to name after the ones we lost.
All of this, it brings us to what might be the deepest question of all. When we talk about bringing a species back, what are we really trying to save? Is a species just its genetic code? Or is it something more? The kinship, the learned culture, the unique way of being in the world that truly defines it?
Just think about the dodo for a second. Here on one side, you see a painting of a living, breathing creature, and on the other, its skeleton. We might, someday, be able to reconstruct its body from its DNA. We can build a machine, but how could we ever possibly reconstruct its way of being? How do you rebuild the ghost in the machine?
A genome, it's like an instruction manual for building the hardware. But it's not the whole story. It doesn't contain the software, the learned behaviors, the social structures, the culture that's passed down from parent to child for generations. A tank of liquid nitrogen can preserve the code, sure, but who's going to teach a revived dodo its song? That knowledge, that's lost forever.
And so, we're left at this huge crossroads with a really fundamental choice to make. Are we truly trying to save the rich, complex, living tapestry of a species, or are we just settling for saving its code in a freezer? This isn't just a question for scientists in a lab. It's a question for all of us about the future we want to build and what we truly value in the natural world around us.